Welcome to this month's Cannabis Research Coalition presentation. My name is Ali Bekhtash. I'm Chief Science Officer at Purple City Genetics. And during this presentation, I want to present to you an introduction to Purple City, both Purple City Genetics and Purple City Labs. And then in the second part, the kind of the meat of the presentation will be about hop latent viroid and how at our nursery we've been able to employ certain methods that we've developed in the lab to deal with this pretty nasty pathogen. I'll start with introducing Purple City Genetics. We are a Oakland, California-based cannabis nursery and breeding company. The official start date is 2008. As a nursery supplying the medical cannabis market of California with cuttings. But of course, the founders and the longtime employees have cannabis experience that go back much longer than 2008. Over the years, it has established itself as one of the mainstays within California cannabis. And we distinguish ourselves in the quality of our product and having a pulse on the market and the market trends, and having our breeding activities match those as much as we can. We both distribute cuttings of varieties that other breeders have selected, but also have an extensive breeding program of our own. This is both in the realm of choosing new individuals to propagate vegetatively and release onto the market, and also over the past 5 years, focusing more and more on seed production for more broadacre farms that are popping up. This, of course, is the trajectory of cannabis in California, a pretty rocky one recently. With the passing of Prop 64, we've observed more and more large farms opening up. Of course, this has kind of predictably been a disaster for legacy cannabis in California. And last year and this year are shaping up to be ones of reckoning to see what the future will look like. But we also recognize that the more that cannabis is subsumed into large-scale agricultural operations, the choice of germplasm will move towards seed rather than cuttings. And also it makes sense when you think about the biology of cannabis, of the species, that it is a seed crop. So feminized, consistent, and stable seed lines will be the dominant type of germplasm on the market in the near future. And this is one of our main focus, is to move the quality that we've established in the realm of cuttings into seed lines. We have different breeding programs with distinct goals, ranging from certain priorities that the farmers have, whether that be pest resistance or flowering time, to the end products of the consumer. And of course, as breeders, our goal is to create a bridge between those, what at times might be differing imperatives. One area that we focus on is our seed line, specifically developed for high-end hash production. It's called El Creme, and it's already started to win some awards at different competitions, such as the Emerald Cup. We get a lot of feedback from different farmers and hash makers and extractors who use our varieties. And the feedback we get has been quite positive, and we partner with different hash makers. The images here that you see of the ice water hash and the butter rosin is from Ati Hash, again in California. So this, I just want to give you a little bit of a taste of Purple City Genetics. There's a lot more that we could go into. We have a R&D research campus in the country outside of Oakland. There's other highlights, but I think the emphasis of this presentation is more about what we do at Purple City Labs, as it's related to the Cannabis Research Coalition's mission. So I'll continue with that. Purple City Labs is initiative that emerged kind of naturally from Purple City Genetics and the various needs. It was created to complement the breeding efforts, assist with challenges faced in the nursery. It provides the nursery with modern agronomic tools, whether that be measuring pollen viability as we're making our crosses, reversing our plants, to counting trichomes for phenotyping, organizing field trials in various places in California and around the world. The organic link between Purple City Genetics and Nursery and Purple City Labs, the R&D focused science company, is, I believe, what sets us apart and makes us stand out, because usually you have one or the other. I mean, of course, there are shining examples of companies that are able to bring both legacy cannabis and a new, more modern scientific approach together. But a lot of times that conversation doesn't happen, or it's just a one-off collaboration. We have many different projects underway. Obviously, we are in the business of developing molecular markers from our mapping populations for different traits that we identify as commercially valuable. We use these within the nursery when we're doing our breeding projects to speed up the work, look at larger populations, move through generations. And another thing that we are excited about is we have an international arm of Purple City Labs based out of Barcelona. And there's actually something similar to the Cannabis Research Coalition there, the Cannabis Hub, which we are a part of. And that's opened up really exciting collaborations with different universities and other companies in Europe. It's interesting when you're working in Europe as a California cannabis germplasm company, how much kind of respect that commands. And then when you also join that with agricultural molecular biology, which of course also has a pretty strong legacy in California and the Bay Area, those two things together make it a fun place to work, being from California. Basically, yeah, we bring a scientific approach to different challenges that face the nursery and our breeding programs. And that brings us to one of those challenges, which I want to talk about today, namely hop latent viroid. I'm almost certain that most of the viewers have heard about hop latent viroid by now. This is very likely not your first presentation that you're watching or hearing about it. There's many podcasts, and a lot of these have happened, in the past 6 months to a year, which is kind of unfortunate because the information had been there much, much longer before that. But I think it took a little bit of time for it to kind of percolate, so to say. You're probably aware, like I said, but I think it's still kind of important to go over some of the history of its identification, where it kind of, the people who put in the work, and how it kind of came to the widespread knowledge that people have about it today. So there had been these symptoms that had been observed for the past 10, 15 years, and mostly on the West Coast, definitely in California, but also in Oregon and Washington, of people reporting increasing numbers of poorly performing plants. And this was colloquially termed duds, just a dud. And it's not really a coincidence, I think, that this emerged at the same time as the medical market drove these clone fairs and clone sales, because it is quite the symptom of vegetative cuttings, and it spreads a lot through people passing cuttings to each other. So the more that activity was happening, the more it started to spread. People didn't know what it was. They were wondering if it's tobacco mosaic virus. There's this idea that is incorrectly termed genetic drift, that if you make a cut over and over again, going through generations of moms, that it loses vigor, which is true, but it doesn't really have to do so much with genetics, but the accumulation of these diseases. Genetic drift, of course, is the change in allele frequencies through different populations that have to do with sexual reproduction. But people basically couldn't figure it out what it was, and in the meantime is spreading pretty heavily, and you just kind of cut your losses and move on. I'd like to acknowledge one of the people I think who has been instrumental in kind of like sounding the alarm, and this was also important for me. I was looking at this issue of a possible virus in cannabis in 2016. I did some work around it, came up empty-handed, and kind of moved on. But then I saw a presentation at the Emerald Scientific Conference in 2017 by Rick Crum from Humboldt Sacred Seeds and Humboldt DNA. And he had collected all of this data up in Northern California, observing these symptoms, making graphs of them. And, he kind of made a very convincing argument that there was what him and John Abrams had termed a putative cannabis infectious agent. So this kind of was the encouragement that I needed to continue looking at this issue. But before we kind of go into like the whole discovery aspect of it, we should talk about what these symptoms are, right? I think a lot of you probably know, but it's still important from an educational perspective to know what we're talking about. So these are two sets of plants, and in each image, the plant on the left is an uninfected plant, and the one on the right is an infected plant. They're the same variety, the same age, grown underneath the same conditions. As much as a controlled experiment can be done, everything is the same except for their hop latent viroid infection status. During vegetative period of cannabis, you see stunted growth, brittle stems. They tend to extend out horizontally in some varieties. The stems snap really easily. Sometimes there's chlorosis, although not always. Smaller leaves, sometimes the leaf blades overlap. And this is all during the vegetative stage, but you can also not observe any symptoms at all in the vegetative stage, and just, especially to an untrained eye, it'll look perfectly fine, healthy plant. Another thing that I think is important, and in this period of the unknown when people didn't know what they were dealing with, became really frustrating in nurseries, is that you tend to get a lot less of rooting efficiency when you're making cuttings from your plants. But then you really notice it once the plants go into flower, right? Huge reduction of trichomes. The flower smells dull, no terpene profiles at all, smaller flowers. Once that happens, you're a few weeks into flowering and you notice this, it's obviously too late. You've put in all that work, and it can be devastating. I mean, some farmers that I would talk with back in 2017, 2018, it would reach such a point that they would just have to start all over again, kill them, start from seed. The symptoms are pretty bad, and an important thing that we'll go into is what I mentioned, is that some plants can be very asymptomatic. So in the vegetative stage especially, but also some of them can ride through the flowering stage. So it's a tricky pathogen. It's not so black and white. So then in 2019, two independent groups doing this research separately identified that the causative agent was hop latent viroid, both in California. RNA sequencing work, basically extracting the RNA from symptomatic plants and non-symptomatic plants and sequencing all the RNA, and then basically looking at databases, public databases, to see if it resembles anything else out there. And the second group, which I wasn't primarily involved with at that time, they also did showed that this was the causative agents by doing basically proving Koch's postulates, where they made a synthetic version of hop latent viroid, infected a plant with it, and then saw the same symptoms. So that is a pretty conclusive way of showing that this is the causative agent. So we should talk a little bit. What is this hop latent viroid? Again, I apologize. I know this has kind of been in the cannabis podcast workshop world for a while, so it might be a little bit of repetition. It's naked RNA. It doesn't have any capsid or co-proteins. We all know what SARS-CoV-2 looks like, right? And it's got the spike protein on the outside. It has none of that. It's just a naked piece of 256 base pairs of RNA. It was identified in 1988 by researchers at the Hops Institute at the Czech Republic, and a lot of the work actually that has gone into hop latent viroid is from the Czech Republic and the Hop Institute there. It can be managed in hops. It's not as devastating as it is to cannabis, and that's a perennial crop. People do see some shifting, reduction of the alpha acids and some terpene preferences towards myrcene in hops, but can be managed. I think the most important thing, as the name implies, that it is latent. So it's not always going to be replicating maybe at the same frequency or causing symptoms. And this allows it to have asymptomatic transmission. The transmission primarily happens through mechanical means. So when you're working with tools, you go from cutting one plant to another one. Maybe there's a wound, there's some sap on your gloves. And that's the primary way that it gets transmitted. But it also, we're fairly certain from our experience in the nursery that it also gets transmitted through runoff from the roots. And there's other viroids, the kind of model viroid species, if you can call it that, is potato spindle tuber viroid. And that one definitely transmits through runoff. And we believe that this one also does. There's this question that comes up about vertical transmission through pollen and seed. I know there's a few groups working on this, and hopefully the results will be published soon. But a lot of anecdotal evidence that there is some level of seed transmission, most likely dependent on the cultivar. Some probably carry it more than others, but it's definitely not a sure shot that if you have seed, it's gonna be not infected. But I'm hopeful that sometime early next year, there'll be some groups that will be able to provide data on that. It's really bad. I can't emphasize that enough. And I don't want to be like alarmist, but as somebody in California, and there's a lot of other states opening up, a lot of other countries, I think it's really important that start your operation with being conscious of this problem, because if you're not, it might be too late. So I think it's important that people who are opening up new facilities, bringing new plants, you kind of want to be careful at those initial stages. what to do about it, right? The most important thing, the kind of goes without saying, not just for hop latent viroid, but for all pathogens, is to have a clean, organized nursery. Disinfect your tools. Nothing's going to beat 10% bleach. It's kind of the only thing that I personally trust. There's all these other products out there that also work and are less corrosive and they don't stain your clothes. But 10% bleach is a sure shot solution. Changing, disinfecting gloves, tracking cuttings so that if you do find a positive plant, you know where it came from. You can go down the line and figure out exactly where the other progeny might be. When you bring in new introductions into your nursery or your grow, you want to make sure that they're isolated. You get to observe them for a while. You're not immediately kind of mixing them in with everything else. And you need to have a very kind of smooth operation, well-trained staff. All of this goes without saying, but I think it's extra important when you are dealing with this particular pathogen. So there's a lot of unfortunate COVID parallels within this topic, and it's actually but you can think about this as wearing a mask, right? It's just immediate. Disinfect tools. It's kind of like wearing a mask against SARS-CoV-2. But then there's another solution, which is testing. This is the only way to, especially in that vegetative stage, even if you do see symptoms, it could be something else, right? A lot of symptoms in plant pathogens can resemble each other. There could be a nutritional deficiency. So the only way to really know for sure is to test for the hop latent viroid RNA with a molecular biology method. All these methods that are available commercially will involve converting the RNA into DNA, and then usually it's followed by PCR. Everybody knows about PCR now, and we probably all had a swab amplified through polymerase chain reaction. And that is, one of the ways that a lot of testing labs are also detecting hop latent viroid RNA. The latency requires repeated testing throughout the lifespan of the plant, as much as you can do until you feel confident. It really depends. Some people suggest every 2 weeks for a month and a half, 3 rounds. But also the sampling is very important because of how the migratory dynamics of the viroid within the sap and the phloem of the plant means that it could be in one part and not the other part. The tissue type is very important. So when you're testing, the quick turnaround time, it really comes into play, because if you're waiting for 2 weeks to get results, then who knows what has happened by then. You kind of need immediate results. And then also high throughput, large scale testing is also important. I think tissue culture is something that comes up a lot in relation to hop latent viroid. Making meristem cultures is one way of cleaning up your plant from this pathogen. It also involves different chemotherapy or thermal therapies of the cultures. It takes a while, but also just to remember that a plant being a tissue culture plant does not necessarily equal that it is hop latent viroid free. The testing aspect even comes into play there, right? Because it's a numbers game. You have to do a lot of meristem culturing, and hopefully one of those or a few of those, you're able to outrun the movement of the viroid, and you have the right treatments, but then you have to keep on testing, right? So tissue culture does not automatically mean viroid free. It needs to be tissue culture in conjunction with a testing scheme. Let's talk about PCR for a second. Backbone of molecular biology, invented in the 1980s in the Bay Area. CDIS Corporation by Carrie Mullis. He got the Nobel Prize for it. And it's the gold standard. It's interesting as a molecular biologist that this almost at this point archaic reaction still the backbone of most things that happen in molecular biology involve PCR in one step or another, but now it's a household term, which is kind of funny. It amplifies a known target sequence by cycling between different temperatures. But relevant to us is that at scale, especially at a scale that would involve testing a plant nursery, it requires a lot of infrastructure, expensive equipment to kind of speed things up, report the results, and also substantial training. All of this means centralization. And so there can be like bottlenecks that arise from this. Minimum probably 3 to 4 hours. There's obviously different versions, different companies that do things different ways, but from sample to result, not even including getting the samples to the lab. And PCR-based testing for Latent Viroid right now is probably $20, $25 to $75 a sample, which I believe is still a lot cheaper than SARS-CoV-2 testing. So this is basically the industry standard, how it is done. And a lot of, different labs are different qualities, and some of them get results in 2 days, some of them get results in 2 weeks, depending on the operation is what you're going to get. So one kind of point that I like to make when I'm talking about this is that in terms of pathogen detection using molecular biology methods, you can't really think about one size fits all approach. The problem and the context of the problem is really gonna speak to what method you're using to deal with it. For example, are you diagnosing an individual, like a patient with COVID, or are you screening a population? There's these other, wastewater, for example, surveillance that happens with SARS-CoV-2. So is your question and what you're trying to do really defines the best method, even if something is, quote, the gold standard. So when we think about Latent Viroid, place like the Nursery of Purple City Genetics, we know that large scale regular testing with quick turnaround is what we needed. And we did the whole PCR thing. We tried different labs. It really did not cut it for us. We thought about just making our own PCR assay, doing that, and quickly that didn't make sense at all, just because of how much it was going to take, how difficult it was going to be to train people in it, etc. Identified all of the needs that an appropriate test for our nursery should be distributed. So we want to do it at the nursery. We don't want to send out samples. We don't want bottlenecks, affordable, easy to administer, and it needed to be something that we could integrate into our production processes. So instead of having a separate testing activity, it needed to be part of the whole nursery management. So really thinking about it like just another thing in our IPM protocols. You spray for this, you test for that. It's just a regular activity that goes on. So this pointed us towards loop-mediated isothermal amplification. It's a different amplification reaction invented in 2000 by Japanese scientists in Aiken. I personally have already had a lot of experience in designing lamp reactions and detection systems. So it was kind of a no-brainer to see if we could come up with something that worked for Latent Viroid. Interestingly, although it had been around for 2 decades, it really got very popular during the pandemic. A lot of people started using it because it made sense to have lamp-based point of care tests and other kinds of tests. And even, some companies that were COVID lamp companies shifted to Latent Viroid when that made more sense for their business. It amplifies the target at a single temperature, so it's isothermal instead of cycling between different temperatures like PCR. It's field friendly. This is basically all of the equipment that you need. There's a lot of devices that integrate all of these different pieces of equipment, some that use, droppers, some that have cell phone readers. There's all kinds of options out there for running lamp reactions. But it's friendly to use in the field. It's easy, affordable. You can scale it up, and it definitely has comparable sensitivity and specificity to PCR. If you know what you're doing when you're designing your reaction, all the different parts of the reaction that fit into each other, whether it's the amplification, the visualization of the results. So if you know what you're doing, you can design an assay that will be comparable to PCR assays. It's very tolerant to inhibitors that might be in your sample. So you can use crude samples, which is kind of a game changer. Things that would inhibit a PCR reaction. The enzyme that catalyzes LAMP will just rip through it. It's around 1 hour from sample to results, and the nature of the reaction means that you produce so much amplification that you can actually see the results with your naked eye. So all of this makes it a really kind of attractive and appropriate option for designing a reaction to be used at a nursery. And we're coming from the nursery perspective, so it's a no-brainer. We're like, okay, we need this. We're gonna make it. So we designed it. We did a ton of validation with other labs, just in-house, and we decided to put it to use. We tested all the new mother plants when we initiated a new cutting, at least 5 cuts per mother plant. And this is what I was mentioning about integrating it into our production process. You make a cut before you put it in the rock wool. You make another little snip of a piece of stem into a test tube. In a few hours, you have the results, so you can kind of quickly remove ones that come out positive and then move through with the other ones, and then you can like retest those. The quick turnaround and the fact that we were doing in-house really allowed us to just go hard, do a ton of tests, and start from like a very kind of clean baseline. We also do these maintenance testing of tables and of plants, of new plants. We learned really quickly that leaf tissue, which a lot of labs recommend, is a pretty suboptimal type of tissue to detect this pathogen. We like stems, we like petioles, we like roots. There's been some good work from Toomey Genetics showing that root tissue is a very nice kind of sample for detecting it from different plants. And the fact that we're doing it in-house allowed us to really track infection rates of different cultivars, to focus our testing efforts, balance it with the forecasting of what we need to produce for our customers, identify plants that maybe have a low level of viroid, but at the same time don't seem to be affected by it that much. Just keeping that in the back of our mind as we're thinking about breeding projects. It just allowed us to ask really interesting scientific questions. After 6 months of this, we got it down to very low, depending on the cultivar, 0 to 1%. And then just kind of moved on to just regular maintenance testing to, if anything comes up, we notice it, we can immediately deal with it before it spreads. And we're a breeding company. We're always bringing new material. We don't have the luxury of just being like, okay, this is it. We're just going to keep these plants. We're never going to bring in a new plant. Everything here is clean. We have to bring in new material. We obviously quarantine it. We test it. But even if you do that, no test is perfect. Every test has a limit of detection. So you need repeated testing, and you kind of need a pretty constant surveillance of your nursery. So because this worked out so much for us, and we've been using it intensively for the past 2 years, and giving feedback to everybody at Purple City Labs from the nursery so that they could develop it and improve the processes, we decided to make an actual commercial product of everything we worked on. We were getting a lot of inquiries from our friend nurseries about it, how we were dealing with it. And it just made sense at that point to make this available to others. We learned a lot of lessons in the past 2 years about sample types, sampling frequencies, creating a more streamlined user experience. So part of our kind of how we make this available is also to consult on best practices and train people on testing on how to do it. There's different formats for equipment and on the volume that you're doing. And we believe that at this point is kind of the most cost-effective and scalable hop latent virus solution on the market. And we made sure to develop it beyond the needs of the testing at Purple City Genetics, so it's as easy and accurate as possible for others. We made different kind of updates to it so that newcomers to molecular biology have an easier time. We did pilots with other places to observe how the users, the workers at the nursery were interacting with it. So we kind of at each step, we felt more confident about launching it. So if you want to give it a try, and we try to make it as accessible to people to just, take it on a test drive, you can get in touch at this address, and somebody will get back to you about making it available. And that is basically all I have. It's kind of a bummer that this is a pre-recorded presentation, because usually I get a lot of questions. It's nice to discuss things with people, but that's my address up there. If you have anything at all on this topic, I would love to connect with you. I'm really excited about the Cannabis Research Coalition. I'd love to try to formulate some projects. So thank you for inviting me. Thank you to the Dr. Allison Justice for getting in touch, and looking forward to next month's presentation.